So there's several versions of the central dogma. But the discovery of RNA splicing and split genes didn't change the central dogma in either version. But what it did change is that the process of taking information from the DNA in the form of RNA was much more complicated in our cells. And that this splicing process, where you remove intronic sequences and make the sense sequence, can vary from cell to cell. In your brain cells, it's done one way for some genes, and then your blood cells another way for some genes. And therefore, it makes the interpretation of what a gene is and how the information or sequence of a gene is then used to make protein much more complex. So though we might or thought to have 23,000 genes, by the time different variations of that gene structure is used to make proteins, we really have hundreds of thousands of different functional units that then carry out the activity of a cell. And therefore, simply knowing the DNA sequence has told us something about the nature of the sequence, but it hasn't told us a lot about how a cell functions. And in that difference is the impact of the genomic sequence on understanding disease processes.